Welcome to this quarter's first Monster Jam seminar. So it's a great pleasure to get to introduce Usha today as the first speaker for this quarter. And Usha's talk is entitled Anticipating Environmental Changes and Front-Loading the Science to Guide Natural Resource Policy, a Leadership Call to Scientists. But before we get to her talk, I would like to give you a brief overview of Usha's journey to and through NOAA by way of a few photos. Usha received her Bachelor of Science degree in Chemistry and Physics from Bombay University in India, and then came to the U.S. to attend graduate school at Caltech, which everybody knows, California Institute of Technology, right? And Usha was only one of 8 women students and the first from India to attend Caltech. So at Caltech, she received her master's in chemistry. And after Caltech, she moved to the University of Washington and received her PhD in chemistry. And then before joining Fisheries here at the center, Usha was a research associate at the Oceanic Institute of Hawaii, but she spent most of her time in Seattle. She didn't get to go to Hawaii that often. Yeah, some, right? And an associate research professor at Seattle University in the Department of Chemistry. And this is a rare sighting of Usha as she was occasionally found in the lab investigating the uptake and state and effects of polycyclic aromatic hydrocarbons in fish. And this research helped pave the way on a number of fronts, including laying the scientific foundation for seafood safety, for assessing seafood safety after an oil spill. Usha then ascended various leadership roles at the center. Don't look at this too carefully. You might recognize a couple people in there. As task manager for the biochemistry program, she then was director of the Environmental Conservation Division, and then became, as you know, science director, where she distinguished herself until she retired. And she was, like I said, a renowned chemist at the center, and then the director. And then she was, I think a little bit too happy when she did retirement life. But, during her career here, she served on many expert committees and science boards and published over 150 articles. And now she's moved on to become, in this happy retirement life, as you can see. She's a member of the Washington State Academy of Sciences. She's a distinguished scholar in residence at the University of Washington's New College of the Environment, and an affiliate professor at the School of Aquatic and Fisheries Science, and at the Department of Chemistry at the University of Washington. And she recently co-taught an environmental law class on science and policy. So some of you met Rao, and Rao's wondering, who retired at the same time, if she has retired at all. But I think she has. She's having a good time. So without further ado, let's welcome Usha and let her give her talk.
Can you hear me? Okay. So I have only 2 hands and more things here. Okay, first of all, if you don't mind, you can just come forward. But if you want to leave in between, and I don't mind if you have to leave in between, but if you like, you can come forward. Otherwise, I'll be speaking to empty chair, which is recently a really good thing, right? One speaks to empty chairs. So, so first of all, I think I am between the barbecue and between you and the barbecue. So I am going to try to give a short talk, but I am not known to give short talks. So be prepared to have a little bit of journey. So first of all, my thanks to John for giving at least part of my talk done, so I can move faster there. And also, it's just so wonderful to be back here. Not that I really missed coming every morning here, and although in retirement I have not gotten A, but I think I have gotten C at least. It's no F. I'm working every quarter, just one quarter I'm doing something new and interesting, but I am mostly doing fun part of it. So it's highly recommended when you are ready. But till then, work hard. And the people I want to thank from the beginning, and then there will be, as we go along the talk, you will see some names. Veera Trainer, of course, for inviting me, and Rohini Paranjpe for staying on it for last year and a half, trying to ask me to come here. Part of it is that I do give some of these talks outside, and when I give these talks outside, the people who come from the center always ask me, why don't I do that one like this in the center so that people can listen to what I say about you all outside. So this is actually the talk about your work, and I've been thinking for last perhaps a decade about why is it that we are always so struggling for funding. And we are right now again at the same stage in a perhaps even more ominous way than we have seen before. And even when we are struggling, we do actually very well to advance the science. And one of the things I've been thinking about is that if we get our science ahead of the game, by thinking and by being innovative, can we at least have a proof of concept done before a crisis happens when we do get a lot of money? It's not that we don't get money, but we get money at a time. We just have to spend so much time responding rather than thinking it out and doing it ahead of time. At least that's my thinking. And so I thought I would share some of my thoughts about that. And maybe I am somewhat more enthusiastic and a positive person and may not be appropriate here right now at this stage of the game, but I feel that I want to show you some ways of getting things done regardless of political or financial pressures. So the kind of science we do is not, we can't just pick and choose whatever we like to do, right? And I'm going to say quite often we and our, but I really mean you and yours, because I'm no more part of you, but I think I'm slightly part of you. So, and before I go further, one person that I truly wanted to thank, and I want to thank him several times and perhaps embarrass him, and that's Tom Home. Several of these talks that I have given during my being the center director, I have had really a pleasure to bounce off ideas, not just create the slides, but how to synthesize the thoughts that we are trying to deliver. And he has been with me on those things forever, I think, and just wanted to say thank you. So the kind of science that we do is, as I said, we can't pick and choose, so we have to remain relevant. And we have to be responsive. As you know, every time a crisis comes, drop everything and move somewhere to do something, and sometimes even move your residence and do something. And I have done it, John has done it, and many of you have done it when the crisis hits. And we have to be also very reliable, because we are in lawsuits all the time, and if it's not reliable, we are going to have a hard time. But things I want to talk about is respected and innovative. Our science, regardless of how quickly it is needed and how badly it is needed and how everybody is calling and said, is it done yet? We still have to publish, and we have to remain true to scientific rigor, and we need to look at the new perspectives. So this quarter, the spring quarter, as John said, I co-taught a class with my longtime colleague, Professor Bill Rogers. He has been asking me for a long time, but I said I cannot do it while I was part of NOAA, but I can do it after a year and a half of being in the university. And we had absolutely a fantastic time. But the main thing, I want to only do those things where I am not just telling everybody what I know, but I am learning something that they know that I don't know. And I was amazed as to how many lawsuits are based on the questioning of best available science. I think many of our laws we know, whether it is ESA, whether it is Magnuson-Stevens, whether it is coastal zone, whatever the laws that people expect us to implement as an agency, the science is actually front and center, although quite often we don't hear about that science is front and center. We don't feel science is front and center. But when I listen to a number of attorneys giving classes in this, and I was part of the law school, part-time law school faculty, that they actually really strongly believe that if we front load the science and we truly have it ready, they will use it. And they will use it, of course, depending on which side of the case they are, but they will use it for sure. So that is a requirement actually for us to provide science way before. Because why is that a requirement? Because our science, as we know, is very slow to mature. I mean, it takes sometimes 2 to 3 years by the time the publication is done. We have peer review, so our underbelly is always exposed with barbs from our colleagues and very sharp comments about what we missed. Nobody ever says, wow, what a great paper, you did such a good job. Here is 5%, you didn't do it. It's the 5% only they send in the comments, right? We are used to it. And I don't know if you guys can see me because I'm kind of hidden behind this, but I mean to be on both sides. But all my equipment is on that side. Okay, so I have to go here, right? So it gets out in open, and when I first became the center director, I was right in the thick of hearing about scientists by name who are maligned by the lawyers because they had already somehow figured out and gotten the peer-reviewed comments, and they're saying, that paper was no good, and these people are doing bad things to us, because our science does affect people's livelihood. And of course, we need to have data, and quite often when we had at least started in this center, we did not have enough resources to have what I call robust data sets. So some of you who came afterwards might feel like, what's the problem? But we did not have many of our programs. We did not have enough data and so the models were quite working on not that much of information. And we continue to evolve, and we have a paradigm shift now and then. And we said, oops, that's not how it was, and now it is changed, and that throws everybody up. Especially when they have just implemented an action. So that shows that we need to be front-loading it so we can give at least some wording to our managers and our partners that we are going to change things, because science is science, and when we get it, and we have to tell them. So that's another reason why we need to make sure that we are way ahead of the curve, while we still have to be responsive. So what I wanted to do is, how do I give you, I can just give a philosophical discussion, but I wanted to give you few examples, few case studies where actually you all have done it. You have front-loaded it either serendipitously or front-loaded it because there were managers or there was government at a certain time, actually thought ahead of the game and said, what if a crisis happens? What if a disaster happens? What is the state of affair before the disaster happens, before a change happens? So we have had some of those examples. And some of it, people actually, we have stepped up really boldly to do that. So these are 5 case studies that I'm going to go through. Some of it, I hope I don't take too much time, but I want to, since this looks like a cozy group, I think I can take a little longer than maybe 45 minutes to do it. People I want to thank here are Mark Myers gave me some really good slides, and actually several of you told me what is happening now, because when I was teaching in the law school, I asked because they were hungry for science, and so I asked some of you, and you were very kind to show me what is happening now. The Vera trainers with harmful algal bloom, Robin Waples and Mike Ford gave me the coast-wide status review and set me right in some of the things. George Pess told me about Elwha. And for oil spill, it is a number of people, but especially Tom has been and Gina have helped me put some things together here. I'm not going to show you a lot of data slides because that's their work. I'm going to show you why it has worked and why under circumstances where we hardly had any staff and with students, how we have done it. And situation is changed quite often, but I don't want you to think that that's the only thing I'm going to do. I'm preparing 3 lectures over period, and these are some of the other topics that I'm not going to spend time, but if you can just quickly look at it, I'm thinking about it. And I think you all know I think about things, and then I will perhaps do something about it. And so these are some of the thoughts that's going through my mind: ocean and human health, increasing carbon dioxide levels, and so many different names we use that people sometimes get confused as to what is this different and how to put it together. Aquaculture has been a challenge and a puzzle and a success. And I think microbial stuff is the most exciting because this center is actually a leader in the agency, but only gets called when there is a crisis, and we need to figure out how to be ahead of the curve. Of course, marine conservation and management is the big kahuna, and that's where front loading happens all the time. You have to because by the law, but we are, I just want to think about how we developed over the years, over the 20 years, and what lessons we learned. Marine mammals, my career, as John said, started with Oceanic Institute, where I did work with marine mammals and echolocation and biochemistry. And I think we have come a full circle when I left with ORCA, recovery planning, and some of the chemistry actually is playing big role in it. So want to think about that. And then I perhaps will get educated about ecosystem approaches. How are they working? I think I spent some years in Puget Sound Partnership Science Panel, and people, and you will see my first couple of slides, ecosystem was a word in mid-70s. And the phrase was, and people are still surprised as to what does that mean, you know. So anyway, we are making major efforts. So how should I thank everybody? I thought, well, I can just thank you all for coming here, listening to me, but also giving me fodder for thinking and keeping my brain cells alive. So this is really wonderful. So this was our last picture together, and before I left, this was done in October 2010 when we did the party for the center. So now I want to talk about chronic pollution in urban estuaries, and I just will talk only a piece of it with hydrocarbon research. And as John said, you know, decades of perseverance, doing things even when we have a lot of challenges. This is exactly what it is. We started in 1975 when I was hired, and the thing that we did, and that is the first time I learned that actually managers do think ahead, because it was a time when you would think, oh, we are now learning strategic planning and all that. They actually, when the Alaska pipeline was being put together and was being laid, the questions were asked in a program called Outer Continental Shelf Ecosystem Assessment Program. And that program was actually set up by the federal government funding, and NOAA managers were asking the question, what if there was a leak? What if there was a bursting of pipes? This is a what if before the event happened, which is practically I don't see nowadays. I see after event happens, oh, where are those scientists and what do they know? Let's find out. Otherwise, is it done yet? But this is a program with which my career started, actually ask those questions ahead of time. And I'm going to do it in two parts. I'm going to talk about the same time in Puget Sound, the chronic pollution with hydrocarbon and other contaminants were being found, and more than that, it's the diseases that were found. So I did not know how to jump from one side to other, so I'm going to show the urban pollution part first, and how some of our science we had done, how it came into being, because nobody knew what the biochemists did in National Marine Fisheries Service. What the heck are they doing? What is a GC mass spec? Kind of stuff at that time. So this is now 20-some years of research and concept put together. And now I must use this. So the most of the money went into asking the question, what is out there, which is the analysis of chemicals in sediment, which was actually marine sediment. All the standards were formed, and this lab here was really a pioneer in trying to work with in the marine sediments. And the last one, and what are the effects? Those two were the bookends, and that's where the money went from outer continental shelf as well as from Puget Sound, some of the funding we are getting. The biologists and pathologists found a number of effects. Main is Mark Myers and Bruce McCain, and a number of people worked on seeing liver tumors in bottom fish. But also in Alaska at the same time, when this OXEP was going on, there was a study done to see, did we have diseases of any kind that were caused by microbes, caused by natural causes, and the environment the fish lived in. So we had a data set that was before any kind of change happened. And then, 10 years later, 15 years later, we had a big oil spill. So we had actually some data from actually Alaska bottom fish. And this side of the equation came in much later because there was a concern in the agency. We don't want to tell people that there can be contaminants in fish, because that is the marketing people, and there was a lot of concern in the industry. So this we did not do at this time when we started in late. So these two questions were important because in Puget Sound, high levels of contamination was being seen, but also effects were being seen. And the tendency to connect those two were very high in the newspapers all the time. In the Alaska side of it, when the oil spill, when there was a question about this pipeline, there were two oil spills, one in Norway and one in England, and they were finding that the fish did not have any hydrocarbons in their tissues. This was in 75 to 77 time frame. And the question was whether fish had very good olfactory mechanisms so that they were just avoiding it because they were not finding the oil in the tissues. When I say oil, I mean hydrocarbons. So I was brought in not exactly to ask these questions, but we asked them, but to look at some of the biochemical side of it. And the question we started to ask is it bioavailable? Because there was one concern, if it goes into marine sediment, the hydrocarbons and compounds like that are too large to be absorbed by fish. And the second was, even if they are absorbed, they have no tendency to do anything with it, so they may excrete them. Third was they avoid it. So all those things were going on, and so we started with this, and John showed one slide of the motley crowd that we had. We had part-time FTEs and temporary FTEs, and we mostly had students. And we asked the question by giving fish a radioactive tracer with the hydrocarbons, and lo and behold, you immediately found large concentrations of radioactivity in the bile. But most of the people who were working on in vitro metabolism using liver enzymes for fish kept saying that the fish have no ability to metabolize because then we found out that they were doing it all at 37 degrees, which is what mammalian bioassays are done with at mammalian temperature. So we, being chemists, we started to do some temperature curve and found actually 25 degrees in vitro metabolism that you could get the maximum conversion of hydrocarbons. So we had in vivo studies that showed that the fish were metabolizing, whether any scientist believes it or not, it was coming out in the gallbladder. But also in vitro, we were able to establish that it happened. Now, we were very under the radar, very small group. Nobody really cared what we were doing at that time. And that was actually a boon. Sometimes it is a boon when nobody knows what you are doing. So you could do quite a lot of it before it found out. So we go forward, and I don't know what's the next slide. Okay, yeah. So this is the conclusion of several years that vertebrates, marine vertebrates, do have very active system to metabolize in the liver and the gallbladder. It's released, and because hydrocarbons have fluorescence, they were easily detectable, but only by really good chemists like Peggy Cran, who was able to develop a method with HPLC and shown, and we use those kind of methods even now. And they are very specific to two-ring compounds to five-ring compounds, naphthalene, phenanthrene, etc. At the same time, we were also looking at what happens, why do fish liver show cancer and tumors, and that was if 10,000 molecules going this way, one of them does get activated and binds to DNA, most of the time gets repaired. But these adducts are, if they are continuously being exposed, is considered the first step leading to mutagenesis and carcinogenesis. So this work, when Mark Myers and all the pathologists were looking at the tumors, that we are beginning to see that actually bottom fish were having, so how to look for whether they are having that system of DNA adducts. This was a particular method found by using labeling of radioactive labeling of DNA after the fact, taking the fish from contaminated areas and hydrolyzing them to a nucleotide, so there is the phosphate groups there, and then you could exchange P32 with a radioactive label. And this work was done, this binding, and this method was developed by Bill Reichert, and all of us involved in it, but he could do it. He knew how to do methods. So we were able to use them. Now these two are, now I'm going to take you very fast now to 1990s. These two are then very good indicators. They are bioindicators of exposure. For the bile shows the indicator of exposure for fish to contaminated area. The adduct shows exposure but also early effect. So these are two very good indicators taking us to the next step. So in 1990s now, we had Eagle Harbor was identified as, and my years may be slightly off, but identified as a Superfund site because there is tremendous amount of creosote stored there and a lot of creosote pollution, so hydrocarbon pollution, and very high levels of liver tumors prevalence. And then in 1993, the capping with clean sediment was done. So actually habitat remediation. And the next slide shows that there was monitoring done practically with not much money because the scientists who worked on these processes and had found these indicators continued to analyze where deal was made with EPA that if they are putting clean sediment, and then we can start looking to see if the levels of contaminants are going down by showing the bile levels. These are the before the these are the risk of toxicopathic lesions before the not totally before, but soon after the clean sediment was put in. So there's a high risk before, and then its risk start to go down. And Mark, just say if I am not saying it right. And the DNA edits were used as another indicator. And you see, the only point I want to make here is that you have to persevere, and you have to take your science, when especially it is very unusual and nobody gets it, to take it to where some action on the ground is happening. And this was an experiment going on the ground with clean sediment being put. We cannot do that kind of experiment. And the fish started to show decline in exposure, decline in DNA edits, decline in the risk of getting tumors. And the most interesting finding to me, because people say, oh, boring, it's going down, so what's a big deal? It worked. So it gave very good immediate information that habitat remediation was successful. But there is a tendency to leave it after a few years. And we start to find, and the scientists are beginning to find that now, after some years in 2010, they are starting to see some change. There is an increase, and Mark just told me last night that there is as much as 17% increase in the risk, right? And so what it's saying is that there is actually a breach somewhere in the cap, or there is a new source of pollution there. And because they are Mark, Jim West, and Sandy O'Neill, who are doing the science and the monitoring, they were immediately able to talk to EPA managers, and I think they are going to be making sure that this remediation remains successful. Okay, so I took quite a lot of time doing this because I thought this is something some of you may not know. Now, the second one is very short, and most of you may know, and that is the copper toxicity and influence of science to change policy. It's very short time, actually, because the scientists were working with salmon, and copper is in the water quite a lot, and is coming from the source is identified as the brake pads, and it was shown by Ned Scholz and his group that actually olfactory and lateral line receptors are damaged. And this data was both credible and have a reliable, so there is a long history of these people doing terrific science, that they were able to communicate it to the legislation, and there are many states have the legislation to actually do source control for copper in the brake pads. That is like, wow, but all the pieces of the attributes of science that I was telling you were used by this team to get to this. So the next one is again, the other one is a short time change. The next one is again a long story, and I'm hoping that timing-wise you will just sit here and listen to me. But this one has a terrific science story with lot of partnerships. And so Vera Trainer helped me put this chronology together because it is a 20-year story. In 1991 was the first outbreak of domoic acid, and Mel Eklund's group were primarily looking at the shellfish industry and looking at, because the Japan had said they will not accept any crabs from United States, especially from West Coast. And so there was a lot of, this is a crisis. We had not planned anything. An outbreak happened and a crisis happened and everything thrown into it. And some methods were developed. But it became very clear that we really, if we wanted to be ahead of the curve, we really needed to start having some intellectual might and some hands on the what you call deck to try to be prepared with understanding of what causes outbreak of domoic acid, what happens with algal blooms that become, why is it happening? We need science, we need monitoring, we need methodology development, and then we need to make sure we have enough partners that are actually our eyes and ears to look at what's happening and get that science converted into public health policy. This is a huge undertaking and require all types of people to be coming in. But the leadership of this center, developed this program where I think Vera Trainer and Jack Wakal were the early leaders in this group to start working on this biotoxin program. I am not going to go through all of it because, again, time is not there, but Olympic region harmful algal bloom is a success story of unusual partners, and that what is most important is the sicknesses are avoided by early warning. But also what is very important is that true science is done. With the initiation sites are identified, the way the oceanographic currents take the harmful algal blooms, the move around, and how can we give a very good early warning all the way down the coast? How is it going up the chain from the phytoplankton when chemical is released, biotoxin, how does it go up the food chain? How do we make sure people truly take this seriously and still don't get so frightened that they don't eat seafood? So we have these two sides of the story, right? And this was done, and so the Olympic region HAB program did all the partnershiping and all. The EcoHab, which is the NSF and NOAA supported funding, did all the science to give the credibility, not to frighten people unnecessarily, but at the time make sure that public health agencies were doing its thing. And I'm not going to be able to show anything more, but these two are two different types of bulletins that were produced. It are now I don't know if it's in our website, Vera, or is it on the state's website. State is a big partner. And now lastly, the Puget Sound Toxin is it called Sound Toxin? You and Rohini and all are involved in that. That we need to look at all the sites and then do triage to make sure that only those sites where we see some problems, that it is we spend the money. And so this is another one of quite an example of front loading. It fell on our lap and it crushed us in the beginning, but then we decided that we needed to be upfront. And it was a very small program, and it still remains, as far as fisheries is concerned, not a very big program. I do not understand, but that's how it is. We don't flail and get upset, we just do it, right? And when I say we, you all. Okay. So, and this is not a small thing. In addition to the health, it is economically a very big impact the state can have. And so partnering with shellfish industry has become as another unusual partners to work with. Okay, so I take you now to a totally different, I've been talking about habitat degradation and remediation and warnings. And now I'm talking about what happens to species when we have this one amazing example. I just came on board in 1994 as the science director, and I found this amazing group of people in those days, coastal zone and estuary forget the last part, yeah. But what was happening, as you see, and this I got from, and I don't know, we just could not get the title clear, Robin, so I don't know where the magic was. We didn't have. So it's a history of West Coast salmon biological reviews. And I only want to show you the two ones in the yellow because I think perhaps I'm far away in time. But we were starting to get petitions, and I think most of you know that when a species to be protected under Endangered Species Act, we usually get a petition from outside, and then Fisheries has 1 year to do the assessment, and then it is decided by the regional office how to, whether to protect it under ESA or not. And we started to get, the scientists were starting to get one by one this petition from 1990 to 1992, and it was like done piecemeal, one at a time, and that was just going to kill us. And so the managers, especially Northwest Regional Office, the attorneys from the General Counsel, and the scientists from this center came together to say, I think we should convince the agency to, and some people from headquarters, I think in protected species, that we should convince to be truly front-loading this information. This is true front-loading because not all the species were being, not all the petitions were coming for every species. So instead of waiting for each one to come, the entire West Coast, the status reviews were done. The assessment of their status, looking at the habitat, looking at their abundance, looking at productivity, their diversity, their genetic fitness, and try to start putting together information that could actually be used. And the information for salmon especially doesn't come only from federal government. Quite a lot of it is tribes and states. And so collecting all this data and then working on it with the status reviews was just an amazing feat for scientists to actually align it. And the regional managers funded us, and we got some funding for it to do that. And people for 5 years, as you see, from 1995 to 1999, many people's lives were totally vested into making sure that every species was analyzed, I should say, every evolutionary significant unit was assessed for whether it needs protection or not. Of the 57, I think, 25 or so, or 27, I'm kind of forgetting, but 50% of those ESUs were considered needing protection under Endangered Species Act. This is like, some people even said, why do you want to poke in your own eye by doing this ahead of time? Let's just wait till they come. But we felt, and especially the scientists and managers, this is our role to be conservation-minded, and this is our role to front-load the science. So this was true front-loading of scientific information that was provided to the managers, and then, as I said, 50% of the ESUs were protected. And once they are protected under ESA, you have to develop recovery plans. So all these recovery domains were they are geographical, they were crafted, and then this is the process that was used. And I don't have so much time, but once again, the Science Center put itself forward with the scientists saying, we will do the technical recovery planning and develop the goals and the conditions in which the fish can come back so that we have robust recovery before the recovery planning is actually turned over to the implementation team. So, and we made sure that the science was still, if you remember, science needs to be respected, so we tried our best to have a panel of scientists that could also give us advice and criticize and tell us whether we are on the track. Now this is another, so I'm doing one very long-term challenge and what we are doing, and one very short term where results are being seen by front loading it. This is George Pess, and I forget the name of that group, called Effective Habitat Remediation or Restoration or some program. But anyway, you know your colleagues, right? And this is the ecological assessment before and after Elwha Dam. I don't think I need to give you a lot of background because it's in the news all the time, but this is a huge experiment happening in our backyard, and we have to be there, although we had no funding for it. The act that does not have any sentence or even a word that says science should be done or ecological assessment should be made. It only says dams will come down and so fish will come back. That's it. But we had to, I mean, truly, I would say, we had to scrape the bottom of funding that we had here in the center to come up with 50K or something very small to make sure we were partners. Because this is, I mean, we know that if you don't have before data, what's the point? I mean, the fish will come back, but we won't be the wiser as to why it worked, what changed, what ecological system, what it changed. And so I am going to just show you, I think, a slide that just says what I just said, that we needed to at least get the samples. We didn't have money to analyze them, but they are in the bank and can be done. And then people are happy, fish are coming back. All kinds of people are very happy that fish are returning, we are doing it. But for scientists, it is this part of thing is that you have to continuously stay with it, monitor, and get as much data so we are smarter the next time when the next dam comes down somewhere. And we have quite a lot of information of how an ecosystem regenerates itself from being totally blasted. This is an absolutely wonderful set of information, and that we have played it. What did I do? Something funny? No. Yeah. Okay. So this is the last one. This is the catastrophic spills and storms. I'm not going to talk about too much, but I have promised my university colleagues that I will tell you that undergraduate students and graduate students are a major resource that we could use. We sit next door. This is a student that I had in 1977, and he worked with me. He always wanted to go. He was from Seattle U, though. He always wanted to be MD PhD, and he knew when he came, he was a junior, he knew he was going to be MD PhD. And we worked, but he wanted to do something interesting and publish a paper if he can. He worked with me for 2 years, and then I'd completely forgotten about this. Still, I gave the seminar at the university, at SAFS, and I looked him up. I just looked him up in Google, and he has not only has MD PhD from Duke University, he is the head of pathology department at University of California, San Francisco. And when I sent him this picture, he told me that this first paper, he said, I learned that long time later, that if he didn't have a publication, he would have gotten into MD program, but he wouldn't have gotten into PhD program. That he feels, and he sent me his CV, and so he wasn't just making me feel better. His CV, the first paper is this one. So remember, when funding is low and staffing is making you very nervous, there is a whole bunch of very eager beaver, very smart youngsters. They will become future scientists if you give them a chance. Okay, so I've done that job. Now, very quickly, the fish, the invertebrates metabolize. We also started to find, but invertebrates don't. So when an oil spill happens, we need to look at, and so we had another experiment in '89 that major oil spill, Exxon Valdez. And of course, everybody was working very hard, just trying to keep their eyebrows over the water line, so to speak, or I should say nose, or we'll drown. But everybody, but all of the scientists here in the Environmental Conservation Division were ready and willing, and other divisions even, ready and willing to go and sample, because what was very important was actually sampling and collecting so that we have some data. But, and we found that what we had done in the lab over the years actually was true, turned out to be that fish showed all kinds of exposure by looking at their gallbladder, but not in their edible tissues. But on the other hand, the molluscan species were showing huge levels of hydrocarbons. Again, I'm just showing you like a, just a quick one. So every time you develop new science, you have to keep your eyes open, or sometimes, it falls on your head, that there may be a way to show that your science actually really was ahead of the time, but was applicable and ready to be applied when it happens. And so these same methods are used over and over again, the same concept over all different types of storms and disasters. My concern, so I'm thinking we do an excellent job in seafood safety, and we are the best, and we have a long-term reliable, we have long-term history of performance. But my concern has been that almost every, and I'm just look at the last three, every major oil spill and many minor that I haven't even given that much thought about, we don't have baseline data. We don't have what was there before, because our focus is only on what's happening now and where the ball is falling. And my science, my colleagues on the science centers told me when I did this front loading, oh, that is suicidal to say to our manager that we should front load. They are not even paying us for responding. But I think unless we front load it, we are always going to be more in the courts and more wondering about, did it really change? I mean, when we have big oil spill and everything is oiled and dying, sure, change. But my long-term effects, did it really change? We don't know. So this is one thing where I am having serious concerns, and I feel that our budget goes up during the crisis, and then it comes down, and we have not really the way to work on it. So I am not going to talk about dispersants, which is a very burning issue for me, that was spilled without us having either the formula or having the method that we had to do it. People came through, they did it, but I don't feel that's the way we should be treated or used. The scientists should be able to ask those questions in between the spills and have a trajectory like it was when OXAP was founded. We should be having trajectories, and people who are taking the oil out of our waters should be asked to pay for it. So anyway, I published a paper. I think I'm getting pretty good reviews. They say it will be 25 years before anything will happen. So I will be gone, I think, for sure. But I hope some of you keep at it, because the science has to be done upfront. So this is what I said. We ought to be getting better, but quite often we can't, because we are moved from one crisis to other. And of course, I just think that public relations sometimes speak for us, which makes it very difficult to get a clear message out. At least that's my thinking. They said experts should not be asked to talk because they know too much. Now that is not a good state of science. So we will continue. We must continue with developing and keeping at it no matter what happens. So here somebody asked me to just say what worked for all of these programs and what worked for me as a scientist and science director. I think risk-taking is a biggie. And I didn't put strategic in front, because I think strategic risk-taking will take out most of the creative risk-taking. So you first say, this is a good risk to take, and then say strategically how to do it. Do you see what I'm saying? The difference here. Staying power, man, I mean, I just gave you several examples of staying power. Innovative funding strategy. All of these programs, case studies that I talked about. Unconventional partnerships. Being a catalyst, sometimes just put a little bit of seed money and give a push, like this series, right? This series we have is just started with practically 3 or 5 of us saying we need to have seminar series, and or anything, just developing any program. And then, like some of you have done, being very close to whether it is Elwha or whether it is Eagle Harbor, whether it is Orhab, being very close and a great communicator. I get quite a lot of compliments even now as to how fantastic center scientists are at communicating when something happens in simple language. Like Einstein says, if you can't explain it simply, you haven't understood it. So we should be able to explain it simply. And I personally feel that to be mindful of diversity. I don't mean diversity only, of course, I mean that too, that it should be gender and color and national origin, culture and all that. But I also mean diversity of disciplines, diversity of approaches, and diversity so that each other of you listening to other people, only people come to a talk which is in their field is not going to, may make us better experts, but it's not going to expand our thinking in an unconventional way. And that's it. Thank you.